Good morning, good afternoon, good evening, depending on your time zone, everyone. I'm Nicolas Loof. I'm an informatics solution leader at BDB Informatic, aka Flojo. I put my email address here, so nicolas.loof@bd.com. Don't hesitate to reach out if you have any question. I also put the support team, flojo@bd.com. If you have any issue with the software, just email the support team. They will be the faster to reply to your question. So in today's webinar, we're going to talk about high-dimensional workflow, and especially the three algorithm FlowCut, PacMAP, and XShift. So here's the outline of today. A brief introduction of Flojo software, platform, and plugin. I'm going to show you how to install the plugin. Then briefly going to talk about the data I'm going to use today for the live demo. And after, we're just going to go to the author of the talk, which is the workflow. So I hope everybody is familiar with the basic of Flojo software. So we have a workspace that allow you to organize your file, annotate your metadata, view your gating hierarchy and statistic. We have the graphical windows, which basically is the heart of Flojo, where you can draw your gate and define your population either through a pseudo color plot, a dot plot, a histogram, whatever form of visualization you choose. Then we have the tool layout, the layout editor, which is your graphical export for publication or presentation, and your table editor, which is your statistical export to create your table. On top of the basic of Flojo, we also have some platform. So we have the composition platform to do your composition or your mixing. We have the export concatenate platform, so it'll allow you to slide or dice your different population, export that to a new FCS file. Then we have the biological platform, which are the cell cycling, the proliferation, the kinetic, the population comparison. And the last platform is the plate editor that allow you to quickly annotate your data, adding meta cluster if you're running your sample in plate. On top of that, we have the plugin. So plugins are extension of the software, a little bit like your app on your phone. So they are developed either by a third-party developer or by the team here at Flojo. So how to install those plugins? So first, where do you find them? Just go in Flojo, go in your workspace tab. You're gonna find in population plugins, and here in the plugins you have the list of all the plugins that's been installed in your Flojo. If you haven't any install, you can click on the top on Flojo Exchange, and it's gonna open the Flojo Exchange web page. If you can go also directly to the Flojo Exchange webpage on typing flojo.com/exchange. So here you're gonna have the list of all the plugins available. So you can select only the plugins available in Flojo, and you're gonna have your list. So we have currently, I believe 67 now. We've been with a few for the past few weeks. So here we have 64, and I think added 3 more since this flyer was made. So in these flyers, you see all the plugins available. So you have the name of the plugins, the color indicate the type of plugins it is. So is it a cleanup plugin? Is it a clustering plugin, a dimensional reduction plugin? What else do we have? Data QC plugins, etc. So you have the name of the plugin. On the bottom right, you have the language it's using. So either R, Java, Python, or C++. And on the top right, you have the output of that plugin. Do you get a new parameter, a new population, a new file, etc. So when you find the plugin you want to install, just click on the download button on the website. It's going to download the plugin as a zip file. You're gonna have to unzip that zip file, and you're gonna have to inside the zip file, you're gonna have a .jar file, and you need to put that .jar file into your plugin folder. So this plugin folder can be located anywhere on your computer. So it can be in your My Document, it can be in your download folder, it can be in your program file. If you are on a PC or your application folder, if you're on Mac, really doesn't matter. As long as you tell FlowJo where that folder is located, then you just have to restart your FlowJo, telling before you after you tell FlowJo where that folder is located, and then all your plugin will be available here now in your plugins in FlowJo, in Workspace plugin. So XShift and PacMAP um, can be installed like that, and also FlowCut. But for FlowCut, you're gonna have to install R as well on your computer, because it's R-based plugin. So to install R, just go on the cron.r-project.org, download the R for your OS system, so either Mac or PC. Then you're also gonna have to install either Xquart or R2. Then you're gonna have to link R to FlowJo by entering the R path into your preference of FlowJo. So just go on the heart logo in FlowJo, go in diagnostic, R path, and put your R path location. If you're on a Mac, it's probably gonna be your username, local, bin R. If you're on a PC, it's gonna be probably your C drive, a program file, or bin, and you're gonna find there. Then restart FlowJo, and it's good to go. For FlowCut, all the packages into R will be downloaded automatically when you're gonna try to use the plugin for the first time. So you don't have to do anything into R. You just need to have R installed on your computer. All right, so let's go talk briefly about the data. So today's data for the live demo is going to be human PBMCs. We have two patients. The sample going to be either stimulated or non-stimulated for these two patients. And really, we are interested in the response of the HLR-DR population with that stimulation. So we did a 13 color staining for intracellular with an intracellular staining, and really because we don't know which cell is going to react to that stimulation, that's the reason we do a high dimensional analysis so that we know exactly which population we're going to react, and we are not biased by doing a manual gate. So let's go to the workflow. So here's the workflow on high parameter analysis. So first is going to be a data QC. Then we're gonna do the annotation. Then we're gonna do the concatenation. Really, those first three steps are really to clean up your sample to make sure everything is ready to run the algorithm. Then we're gonna run the algorithm, your dimensional reduction algorithm, and your clustering algorithm. And then you as a scientist go back into play, because now you're gonna have to define every cluster. So putting a biological name. So is it my Treg or my monocyte or granulocyte or whatever cell population you're looking at? And after, because you did your experiment, the last step is going to be the comparison between your two conditions. So for today's condition, it's going to be no stimulated, but for you might be a different time point, or healthy versus disease patient. So let's go in this order now. So you're always going to have this pictogram here on top that reminds you the different steps. So right now we are in the data QC steps, and the first data QC step is going to be our composition. So we need to make sure our samples are well composited. So you have many ways to do your composition. You can, of course, do your composition or mixing on the instrument. And when you import your data in FlowJo, FlowJo is going to be able to read that composition that you've done on the instrument, and it's going to be the acquisition defined in FlowJo. Or you can do your composition within FlowJo. We have different methods available for composition or mixing in FlowJo. So we have the traditional composition, we have the auto spill composition, we have the spectral, which is our mixing. You can remove the autofluorescence as well within FlowJo. You can compare your different matrices to see which composition method worked the best on your sample. Then also on top of all those composition method in FlowJo, you can calculate the spillover spread matrix and the total spread matrix, which is gonna help you for your panel design. And also now in FlowJo, you can calculate your cosine similarity matrix to know if two fluorochrome are really similar to each other based on your for your instruments, or if they are really separate. So really different. So if the value is really close to 1, that means the two fluorochrome gonna be really, really similar, or really look exactly the same for your instrument. If this value is close to 0, that means those two fluorochrome are really have a different spectral signature of one from the other. All right, still in the dialog QC. Now the step is the transformation. So when you display your data for the first time, your data may look squished. So either you're gonna have your way to zoom out, so really you don't see really well the separation between your negative population and positive population, or the other way, your way to zoom in, and you're gonna have a lot of cells on the axis. This transformation step is usually overlooked by many users, but it's really important because the algorithm for your dimensional reduction or clustering really gonna look at the data like you in with your own eyes on a dot plot or a pseudo color plot. So really having the best separation visually also is going to help the algorithm to really cluster or do the different island for the dimensional reduction as good as possible. So spend some time here, click on the T button, click on the customize access, and adjust your scale to make sure that the data look as good as possible there before going to the next step. Still in the data QC, now we're going to do the cleanup step. So the cleanup step is removing any problematic event that's been caused by the instrument. So it can be a clog or air bubble or any instability into the signal. So, and we're gonna also want to remove any unwanted events, so debris, doublet, dead cells, lineage positive cells as well. So for the problematic event caused by the instrument, of course, you can do this manually by drawing a gate using the time gate parameter and removing any unstable part of the run. But I highly recommend you to use an algorithm because it's going to save you time and it's going to do a way better job than you removing any problematic event. So in FlowJo, we currently have 4 algorithms for that. We have FlowAI, Peacock, FlowClean, and FlowCut. And today we're gonna focus on FlowCut. So any algorithm is gonna inspect the FCS file. They're gonna remove anomalous event, and they're gonna return only the high quality event. And it's gonna create 2 populations: the good event and the bad event. And you're gonna keep using the good event population to keep going on your analysis. All right, so let's jump into FlowCut and how it works. So FlowCut has been published. Here's the name of the paper, FlowCut, an R package for automated removal of outline event and a flagging of file based on time versus fluorescence analysis. It was written by Justin Mescas, so in Cytometry Parter. So if you're interested in reading the paper, it's there. So I'm gonna try to summarize this paper as fast, but so as good as possible. So First step, FlowCut is gonna remove the cells that contain a segment with low density or that are substantially different from the rest of the segment across the time of collection. So the first step of FlowCut is really to create really small time bin. So in each time bin, it's gonna look at the number of cells. If you see that all the other time bin that have a lot of cells, but this time bin has really almost like no event, is just gonna remove that time bin saying, oh, you had a clog at that part, so at this time bin is not a good time bin. He also is gonna evaluate the data using Earth measurement for each time bin or each segment. It's gonna look at the mean fluorescence intensity, the median fluorescence intensity is gonna look at the 5th, the 20th, the 80th, and the 95 percentile. It's gonna also look at the second moment and the third moment. So the second moment is the variation of the signal, and the third moment is the squeeness of the signal. It's going to do the density of the summation of the 8 measurement and the 2 parameter using the max valley height and the max per rocket. And it will determine which events are significantly different, and those events will be removed. Additionally, the file is going to be evaluated for this fault check. It's going to be the not monetarily increase in time. So is this signal stable, or is it increasing slightly over time? It's going to look at the sudden change in fluorescence. It's going to look at the larger gradual change of fluorescence in all channels. And it's going to look for very large gradual change of fluorescence in one channel. So let's look a little bit into the report that FlowCut is gonna give you. So first, it's gonna produce those image, and this image on the top is gonna contain 4 letters, and this 4 letters is gonna be either T or a F, indicated whether the sample passed or failed for each test. And the order of the letter follow the order of the test I just mentioned. So here in this case, we have all T's for the full test, so everything was true, so everything passed. Also, on the top of any plot, you're gonna have a series of number. The first number indicate the mean drift before the cleaning. The second number represent the mean drift after the cleaning. And the third number in parentheses represent the max one-step change after cutting. So the second also of the last drop here is going to be the worst channel over time. So between all the channel that you run, the second to the last is the one that has the most issue. So like that is showing you, and you can look into that channel. And the last plot is the density summon measurement. So it's showing the sum of the 8 metrics that FlowCut evaluate. So mean, median, 5th, 20th, 80th, and 95, etc. And the line right there is the cut. So any event on the right side of this line is going to be the bad event. Every event on the left side of that cut is going to be the good event. So let's look into not so good a sample and the report, and see really a little bit more in detail. So here we can see the 4 letters. So T, F, F, F. So that means the first test is true. So it passed. So no monotonical increase in time, it passed. But everything else, it failed. So it fell in the sudden change in fluorescence. It fell in the larger gradual change of fluorescence in all channel, and it fell in the very large gradual change of fluorescence in one channel. So now we have the image display for each parameter. And visually, we can see some spike or drop that indicate anomaly that we should remove or should be removed by FlowCut. So everything that's in gray, okay, or gray or black, I don't know, depending on the color you have on your screen, but everything here kind of in the dark is event that been labeled as bad event by FlowCut and what's been removed. Also on the top and the bottom, you have your 98th percentile and your second percentile. So before cleaning and after cleaning. So the light brown is before cleaning. Your dark brown is after cleaning. And usually, or often, those two lines really gonna overlap. You can see here, we can almost see the two colors. So we see the light brown a little bit on top and the brown line a little bit at the bottom. But for example, on this one, the light brown and dark brown overlap, but you can see on the second percentile, the two are slightly separated. The brown line here in the middle is the mean of each time segment. When the bright lines turn pink, like here at the beginning, that indicate that that segment has been removed by the FlowCut algorithm. Okay, so when it's brown, that means it's kept. Every time you see it pink, that's part that's been removed. All right, so now that we remove all the problematic events coming from the instrument, we will want to remove any unwanted event. So we're just gonna remove the debris by just doing a cleanup gauge with our forward scatter, side scatter to select our cells. We're gonna remove the doublet with a forward scatter A versus H, followed by side scatter A versus H, and we're gonna remove dead cells with a live dead marker, and if you have a lineage marker or the lineage positive. And you can stop there and start doing UI parameter analysis. Or you can keep going to really the sub population that we're interested in to get more resolution. So here's an example where we stop only on the local site, so CD45 positive cells, and we can see our granular site, CD4, CD8 population, B population, and K and monocyte. So we can see all the different population, but if we look at the CD8 population, we don't have much resolution. We see probably like 4 or 5 different islands, so potentially 4 or 5 different CD8 population. But when we start removing any unwanted population, so the granular side with CD15, monocyte with CD14, B cell with CD19, CD20, the NK with CD56, and the CD4 T cells with the CD4 marker. Now we're only looking at the CD8 population, and we can see we get way more resolution into the CD8 population because we can probably see like 10 to well, maybe even 15 different islands, so potentially 15 different CD8 population. Really, where you stop is on what resolution you want for that specific population. You may have to do both. You know, maybe you have no idea on how your cell's going to react to your treatment or your stimulation, and you're going to do on all the cells to see which cells are reacting to that stimulation, and we do the analysis after you discover that was only your CD8 that react to a stimulation. So you want to have a better resolution on all your different CD8 population. All right, so we're done with the data QC steps. So really a big steps here. So take your time on this data QC steps. You need to make sure that your data is clean. Doesn't matter how good is the algorithm you're gonna use after for your dimensional reduction and clustering. If you put junk in it, you get junk at the end. So spend a lot of time there. Second step is going to be the your annotation. So annotation is your keyword. So keyword are metadata that are included into your FCS file. These metadata can be added either on the acquisition software or into FlowJo. So even if I'm a FlowJo person, I highly recommend you to add those metadata on the acquisition software. Because if you add those metadata on the acquisition software, those metadata will be embedded into your FCS file when you export your data out of the acquisition software. So if you share this FCS file with anybody, those person will be able to see the all the keyword that you added. If you add those keywords into a FlowJo, those keywords will only be present into FlowJo. And it's just because at FlowJo we decided that we didn't want to transform or modify your raw data file. Your raw data files are there. We don't want to touch it. We're just gonna read it. And if you want to add information, metadata, we're just gonna add them only in FlowJo. We're not gonna transform your raw data file. But that means also if you want to share those information, those metadata that you created or those keywords that you created into FlowJo with someone else, you're gonna have to share your FCS file, but also your FlowJo file for that person to be able to see those keywords that you added into FlowJo. So that's how to add a keyword into FlowJo. So if you're running plate, you can add those keywords using the plate editor. And if you're running tubes, you have the add keyword button that's gonna add an additional column in the FlowJo workspace, and you can put any information you want for each sample. Step 3 is your concatenation step. And your concatenation step is really the primary difference between your standard analysis and your high-dimensional workflow analysis. So all the steps, the data QC annotation is step that hopefully you already do even for your manual gating. But here really this concatenation step is one step that is new. And what we're gonna do here in this step, we're just gonna concatenate all the sample into one big file. And the reason we do that is because of the nature of the algorithm. And the nature of the algorithm is for dimensional reduction or clustering, the first step is seeding. And this first step is random. So if you're talking about dimensional reduction, maybe you put your CD4 on the top right for the run. But if you rerun the same algorithm on the same sample, maybe for the next run, your CD4 population will be on the bottom left, so on the opposite side. You're still going to define all your different cell population the same way, but the location of those different population in your dimensional reduction, for example, it's not going to be the same. For a clustering algorithm, you cluster one. For the run number one, may not be the same cells that for a different run. So you're still gonna have the same, or at least similar number of cluster, but the cluster number may change. So to be able to compare sample to sample, the best way is to concatenate all your sample into one big file like that. All your cells for all your samples are located at the same place, so it's going to be way easier to compare between sample or between condition. Usually with this concatenation step, we combine it with a downsample step. And this downsample step is not necessary to decrease the number of events, but just to make sure that we have the same number of events between each condition, because we don't want the algorithm to be overpowered by one condition. So we want to have kind of similar number of events for each condition. All right, so that's it. We're done with the three first cleanup step. So now we're gonna go to our algorithm. So dimensional reduction and clustering. So let's start with dimensional reduction. The goal of those algorithms is to be able to represent your high parameter or high dimensional data set to a two-dimensional plot. But also we want to preserve the overall structure of the data as much as possible. So we want to really be able to separate the different cell population. So here I overlay manual getting, and really when we do dimensional reduction, we want cells that are really similar to each other to combine into a small island. So we can see that all the monocytes here group together and create this island. Same thing for the NK, same thing for the CD8, CD4, and the B cells. I see a question. Um, So there's a question on how can you determine which cleaning data algorithm is the most suitable for your data? What criteria should be considered? Um, so, well, as I said, we have four cleaning algorithm right now. So FlowClean, FlowAI, Peacock, and FlowCut. So um, FlowClean was the first algorithm that's been released in 2016. It really was a good first step in the progress of cleaning our sample. Now is, in my opinion, outdated compared to the other one. So I will put FlowClean a little bit on the side. If you use FlowClean in the previous data analysis and is a longitudinal study, keep using FlowClean because like that, you're consistent in your analysis. But if you're starting new today, just probably pick another one. Now between FlowAI, Peacock, and FlowCut, so it really depends on the if you want to do the analysis and the cleanup on the raw data or on the compensated data. So FlowAI is on raw data. So just out of the instrument, don't need to do compensation or anything. You just can run FlowAI compared to Peacock, and FlowCut is on compensated data and even on the transform data. So make sure that your data is well compensated and make sure that your visualization is good, especially for FlowCut. Sorry, for Peacock. After just a different method. So when they've been compared, there's some comparison. They really are similar from each other in the quality output. So they all remove the bad event. Some remove a little bit more of the good event than the other one, depending of how you tweak them, strict you are with those events. But Peacock, FlowCut, and FlowAI really perform about the same level to remove the bad event and good event. So after it just personal preference. All right, so back on dimensional reduction. So now that we understand what dimensional reduction is, so let's look at the different dimensional reduction available in FlowJo. So we have a t-SNE available, we have UMAP available, we have TriMAP, EmbedSum, PacMAP, and Fate. Those 6 different dimensional reduction tools allow you to create 8 different visualization, which are t-SNE, UMAP, TriMAP, EmbedSum. But EmbedSum can do an EmbedSum t-SNE, an EmbedSum UMAP, and of course PacMap and Fate. And if you look, we overlay the manual getting, and you can see that each different population will produce a nice island where the cells that are similar to each other are really grouped together. So today we're gonna focus on PacMap. So PacMap stands for Pairwise Control Manifold Approximation and Projection. Here is the paper. understanding how the dimensional reduction tool works, and empirical approach to deciphering Disney UMAP, TriMAP, and PacMAP for data visualization, and it was written by Xing-Feng Wang. There's another question. Sorry, messed up. For example, if you are using 20 marker for a panel, should all marker be selected when running Disney or UMAP? Additionally, I can redetermine the number of cluster and what step I can retake to reduce the number of cluster created after running Disney. I'm gonna answer that after because that's part of what we're gonna talk here on the clustering. We're not there yet for the clustering. You're talking about Disney or UMAP in the dimensional reduction. You should select all the marker. Yes, maybe except your live dead marker because you already and your lineage if you already get those out. So every marker that require to define your populations need to be selected. So um, the live dead and the lineage, if you have a lineage, if you get those cells out already when you created your concatenate population, you can remove those. And for the following question on clustering, I'll answer that after. All right, so PacMAP, why did I pick PacMAP today and not Disney or UMAP, which are usually the most used in the field, is because PacMAP is claiming to be the best in both worlds. Usually when you pick a dimensional reduction, you either pick, for example, Disney, which is better to keep the local structure of your data, or you pick another dimensional reduction, which is usually better to keep the global structure of the data, like, for example, a TriMAP. And PacMAP here is um, claim or show that is able to keep both. So really nice local structure preserve, but also the best global structure preserve. So let's dig a little bit in their paper and explain why they say they're the best of the two worlds. So the first experiment they did for PacMap to prove that it was good at keeping the global structure of the data, they used the Mammoth dataset. And this Mammoth dataset, as you see here, is when you look at the data in the 3D, looks like a mammoth. Okay. And after they run this data set using either Disney, UMAP, TriMAP, and PacMap, and you see that on Disney, he really like separate the different parts of the mammoth, okay, the different legs, the ribs, cage, the head, uh, etc., into the different population. But really, we don't get the global structure. So we don't really get back the shape of the mammoth. The best thing we did when we moved the perplexity, which is probably for Disney, the most important parameter controlling that visualization, we see that when we change the perplexity, the best thing that we can get is more like a chicken than a mammoth. So really good to separate the different population, but not really good to give us the global shape of those population. So that's why we say Disney usually is not really good at keep the global structure of the data. When we look at UMAP, same thing. We can see the different population. We can see the two legs. We can see really well the toe of the legs in this case, but really doesn't look much like a mammoth in any case when we change the number of the n number of neighbor. Yeah. Compared to TriMap, TriMap is an algorithm that is good to keep the global structure of the data. And here we see that for TriMap, we can see doesn't matter if we change or not the inliers, we can really see that we have a mammoth shape, but not really the local structure, because if you look at the feet of the mammoth, we cannot really see the toe of the mammoth. In comparison, when we run this data set on a PacMAP, we can really get back that mammoth shape. So really looking really similar to TriMap or to the original. But also you can see on the leg here at the bottom, we can really see the toe. So really that showing us that it can really get even better resolution in a small difference of population. All right, so that was for the global structure. Now let's look at the local structure of the data. So for the local structure of the data in the paper, they use the MNIST data set. And this data set to see if we keep well the local structure of data set. If you have a good separation between the different means class, that means you preserve the data set better. So you can see that on Disney, we can really see like nice separation between each different cell population or classes in this case. So everything is good. So really Disney preserve well the local structure of the data. UMAP, same, is not doing a bad job there at all. So we can really see well separated population. So good there as well to keep the local structure. But for TriMap, you see that it's having a little bit hard time to separate the different cell population. And that TriMap we know is really good to keep the global structure, but not as good to keep the local structure. But if we look at PacMap, PacMap is performing at least as well as UMAP, really separating well the different cell population or classes here in this means data. So good as well to preserve the local structure. So PacMap, interesting tool. I encourage you to look at it. It's not, as I said, not the most used currently in the field, but seems really promising to keep the local and global structure of your data. So now that we've done the dimensional reduction, let's do the clustering. So really, dimensional reduction and clustering really go hand in hand. So one is not really useful without the other one. So here I've overlaid the output of a flow sum into a UMAP to really help identify the different subsets. And the reason we're gonna use a clustering algorithm after dimensional reduction is just to be able to crush the data faster. And this clustering algorithm is gonna help us identify all the different population present in the data set. Usually in a few minutes, and instead of just trying to do just manual gating to define those population. So we have three different kind of clustering available in the field. So they are the supervised clustering algorithm, the unsupervised clustering algorithm, and the in-between clustering algorithm. So the one we're interested in when we do high parameter analysis are the unsupervised clustering algorithm, and that's the one I'm gonna present to you today. Know that supervised and in between are also available in FlowJo, but they are not in the scope of what we need today, so I'm not gonna talk about those. So the unsupervised clustering algorithm so is a type of algorithm that learn the pattern from your untagged data. And the hope is that through mimicry, which is an important mode of learning in people, the machine is forced to build a compact internal representation of its word and then generate imaging content from it. We have a 4 unsupervised clustering algorithm available in FlowJo. We have XShift, Phenograph, FlowSum, and T-Rex. And today we're gonna focus only on XShift. So XShift has been published here, so automated mapping of a phenotype space with a single cell data, written by Nicolas Loof, and it was a Nature method in 2016. So XShift is a clustering algorithm that uses the k-nearest neighbor density estimate to find the cell population in multi-parametric single cell data. And the big advantage of XShift is that is automatically gonna determine your optimal Q value. Of course, you can change this K value if you increase that K value, you're gonna under cluster. If you decrease that K value, you're gonna over cluster. So if you're not really happy with the number of cluster you get, and you know that you have more population that as what XShift found, you can just manually adjust the K value, but really normally you don't have to adjust this K value. XShift automatically gonna find that that number for you. And also XShift is a really well fitted to find small or rare population, which is usually what we do in flow cytometry. So that's why I wanted to present that algorithm to you today. So how does XShift work? So first, he's gonna construct the nearest neighbor graph that captured the phenotypic similarity of high-dimensional data. Then it's going to use the distance of the key nearest neighbor to compute local density estimation for each point. And then finally, it's going to search for local density maxima in the KNN graph and apply density gradient ascent to create distinct population. So how are you going to run that into FlowJo? You're going to select your parameter. So all your fluorescence parameter, maybe minus your lifetime marker if you already get it out, and maybe a lineage marker if you already get it out. And after you automatically gonna find the best K value for you, so you don't need to change it. As I mentioned, you can if you want. Then we're gonna be able to change the distance metric. So it can be either Euclidean or angular. Angular is gonna be recommended if you use more than 10 parameters. So usually when we do high parameter analysis, it's always gonna be angular because we're gonna do more than 10. Then we have the sub-sampling limit, which is going to be the number of cells used for the clustering. So almost like a small downsampling there just to run that algorithm. And after you can assign a specific run ID to know which one you use, so like that you can run different time the XShift with different settings. So for example, if you're changing the K number. All right, so we did our algorithm, dimensional reduction and clustering. So now let's look at the data. So in the phenotyping step, because we assign annotation, and really that was the whole goal of annotation here in FlowJo, is we're gonna transform those keywords into a parameter in FlowJo, now we can gate based on this parameter after we did our dimensional reduction and clustering. So here, this keyword tissue where we have our lymph node, blood, spleen, and liver, we can separate back or concatenate file into each independent type of tissue or sample. And then we can look at the differences for each different type of tissue. So we can see, for example, here the cluster on top is really there into the spleen, as well in the liver, not so much in the blood, and totally gone in the lymph node. So we see that this island specifically is changing based on the tissue type. So maybe we want to know which cell types are those. But right now we doesn't tell you which cell type. So we need to know which cell types it is. We're gonna use Cluster Explorer, and Cluster Explorer is not built in FlowJo. So if you go in your population pane, now you're gonna see Cluster Explorer, and Cluster Explorer is gonna give you a graph line, your frequency of your cluster, so either the actual number of cells or the percentage, your dimensional reduction tool, so PacMAP for today, but it can be your Tisney or UMAP, and it's gonna give you also the heat map with your cluster number and your parameter to see the expression of that marker. In Cluster Explorer, you're gonna be able to select each cluster to see the expression into the graph line, so you know which marker are expressed. But you can also gonna be able to gate based on each graph line. So you can gonna be able to say, I want my sorry, I cannot read it. CD56 positive, the CD8 positive, but not the CD14 positive, and Cluster Explorer is going to give you every cluster that match those criteria. And after you can look with your heat map or your graph line, what's the difference between each of those clusters, and rename this cluster with like a biological name. So, okay, activated monocyte, non-activated monocytes, for example, or my Tregs or whatever cell population you have. So that's a manual way to define all your cluster. You can use a more automatic way, which is MEM, which stands for Marker Enrichment Modeling. So MEM is going to compare one cluster with a population of reference. So this population of reference can be all the other cluster, the average of all the other cluster, or can be a user-defined population. And it's going to give you a marker of positivity or negativity, okay, so a plus or minus sign, and it's gonna give you a vector. So a vector going from 0 to 10, so 10 really positive, or with a minus really negative compared to the population of reference, and 0 the same. So if we look at one of these clusters, for example, MEM gave us this label, so HLR-DR positive, vector of positive with a vector of 10, so really, really bright for HLR-DR. Then the CD20 positive, a vector of 9, so really bright for CD20. CD19 positive, vector of 7, so bright for CD19. IgGM positive, a vector of 5, so we're getting in the mid here. CD34 positive, vector of 3. CD45 error, a positive vector of 3, CXCR4 positive vector of 2, CD47 positive vector of 2, CD33 positive vector of 2, and it gave us a CD7 a negative with a vector of 2. That means our population of reference was probably CD7 DIM positive. Okay, with that label, I can tell that this population with those markers are my B cells, okay, CD19 positive cells. So how do you do MEM in FlowJo? Just select your cluster, click on in your population band, and next to Cluster Explorer, you're gonna see MEM and create a MEM score. And when you click on create MEM score, it's gonna give you the list of your marker with the marker of the positivity or negativity and the vector. This list might be huge depending on how many marker you have in your experiment. So you can apply a threshold to decrease that size. So let's say if it's not at least a vector of 2, I don't want to see it, and allow you to have a list of marker that's more readable and allow you to define your different cluster. And so in this example, for example, here we see that my cluster 2 is CD5 negative with vector of 3, CD38 positive with vector of 2, and same thing for my cluster 8 and my cluster 9. They have the same MEM score. So what I could do is just telling MEM to merge every cluster that have the exact same MEM score. And now my cluster 2, 8, 9, which are CD5 negative vector of 3 and CD38 positive vector of 2 have been merged here, okay, in that cluster. So easy way to combine all those clusters if you have a cluster. All right, so now we're on the last step, which is our comparison between our different conditions. So now we want to know if one cluster is overexpressed in this condition compared to the other one. So we can do that in a cluster explorer, or you can do that in the visualization of your dimensional reduction based on your condition. So you can visualize your dimensional reduction. Here is a T-SNP, but it can be your PacMAP, and for your different type of tissue or different condition. So we can see our blood, spleen, liver, and lymph node, and we can see some clustering or at least island disappearing and appearing. Or you can also do on Cluster Explorer, you can add additional column which are based on your condition, so no steam and steam. And based on the color here is telling you that my population 0 is considered mostly from cells coming from my no steam sample, and almost none of them are coming from my steam sample. And the other way around here for population 6 to 14, those clusters are created mostly from cells coming from my STEAM sample, and almost none of those cells coming from my no STEAM sample. And if it's green, that usually means about close to 50-50. So cells coming from both conditions. So no change or really little change between each condition. All right, so we did our full workflow. Okay, so now that you understand the full workflow, let's jump into the software and I'll show you how to do it live. All right, the screen. Let's open my workspace. All right, so I created a workspace with my old data. So two patient 1, patient 2, those teams, team. Okay, so I combine them into a data group so that easier, nicer to organize. I have a compensation already applied to the sample. So really, if we go back to step 1, which is um, data QC, let's look at the compensation first. Okay, so I'm just gonna double click on it. And there you go. I can check my composition right there on my sample, increase so that we can see all the n, almost all the n by n, and I can remove the before composition overlay, which is the cyan. Now the black is after composition, and I can see all my composition is square, so everything looks good. My composition seems to be correct. So my composition is done. We're good. So now the next step is my transformation. So I'm gonna open my graph windows, and I'm gonna start looking at every parameter and adjust my scale to fit for my sample. So I can go sample to sample just to make sure I don't cut it, and I can adjust my scale saying, okay, here I can cut it to have better resolution. There you go. Go to HLRDR. This one looks fine. My live dead, CD1C. Okay, so probably can cut there. So I just maybe decrease the compression, add additional decade. There you go. And right there. Okay. Do the same for TNF. Okay. That looks good. CD83. That looks fine. If I remember correctly, the only one I have to adjust after is just my CD123 here in my list. So let's go directly to here, CD123. See that my positive population is right there. A lot of white space. I can remove that white space. So adjusting. And and there you go. Okay. So I did adjust my transformation, make sure my data looks good for every parameter. All right, so now let's talk about the cleanup. So the circle here is your data QC indicator. So we can go in my tools, Atometry, check sample quality, and I can look at the color of my circle to see if my sample is good or not. So everything here, it's blue. So if it's green, that means you have almost no flaws on your sample, or really little flaws. If it's blue, it's okay. And if it's any other color, so orange, pink, or red, that means you had issue running those samples on the instrument. So here in this case, blue, so that means little flaws to no flaws on our sample. But we're still gonna run FlowCut. So I'm just gonna go in my workspace tab, plug in, and look for my FlowCut plugin. Okay. I'm gonna select my parameter. So my forward scalar, side scalar parameter, and all my fluorescence parameter, I'll select it. Okay. I can adjust the input max or min. Okay, and if you put your mouse over, we explain exactly what it is. So that's the threshold of the average mean drift with this single channel with the highest range of means. So decreasing this value will increase the how strict you are for for the algorithm. Then also you have your input mean or mean, you have your input max content, you have your input max per cut. So all those values can be adjusted. You can manually set the get line force. So you see the cut. Okay. You can use only the worst channel. So if you don't look at all the channel, but just the worst one, and just for troubleshooting purposes, you can set the R script as an output message. And you can also, if you want, add all the QC images that I show you into your layout so that you can really see it. If you don't, not worry, it's available also on in your in the folder. So just gonna leave all the default and just click on run. Yeah. So FlowCut is gonna run for this sample. May take about 20 to 30 seconds. And it's gonna give me the good event and bad event. And you see where my workspace has been created here on my desktop. He automatically created also a folder with this exact same name as my workspace. And within that folder, I have a FlowCut folder where I have the name of my sample that I'm running my FlowCut, so my TLR Steam patient 2. And within, I'm gonna have my output when it's done running. Okay. So if you didn't save those images into your layout editor, you're gonna be able to find your report images here in that folder. All right, so FlowCut is done. Keep good event 100%, bad event 0%. So everything is good. You didn't find any bad event in this sample. Guys. So now we can run this FlowCut. So you can, if you disagree, or you can change those settings. But if you're fine with FlowCut, you can just click and drag your FlowCut node on all on your group, so it will run on all your samples. Okay, and if we go back here on the Minus Team FlowCut, you can see my PNG image, which is my report. So I pass the 4 tests, they're all true. And if we want to see the image, we open the PNG and we can see that everything seems stable. Okay, so when we look at our um, median line here, didn't put anything in pink, so it kept everything there. Okay. All right, so now when it's still calculating for the other event, we're gonna select the good event. Okay, I'm gonna select the good event, and I'm gonna do my manual gating. So my forward scalar, side scalar to select my cells of interest. So my cells of interest are those. We're looking at the HLRDL cell population. So those are my cells. Okay, then for my cells, I'm gonna remove my doublet. Okay. Then same thing for my size scaler to try to remove as many doublet as possible. Okay. And now we're gonna go to our live dead marker and our lineage, and we're gonna select the double positive in this case. So this one is live lineage neg. Okay. So I did 2 sample already, just missing 1. All right, so quick tip. You have all those graph windows open. If you want to close them all, just go in your file, close all graph windows, or you can use the shortcuts. So in Mac is the Apple key Shift and W. In a PC is your Control, Shift, and W. And it will close all your graph windows. All right, so I did run on all my sample. I don't need my FlowCut node anymore, so I don't want FlowCut to run by accident. So I'm just gonna select, delete entirely. It kept my FlowCut derived parameter and my good and bad event from FlowCut. So now I can select good event, bad event, and all my manual gates, and I can click and drag those on my group. So let you apply this analysis to all my sample. And Flojo will be smart enough to know that the good event and bad event from FlowCut are coming from your run. So it's gonna keep it the right values. All right, so now I need to do my, so we're done, sorry, we're done with the cleaning. So now we are on the annotation step. Okay, so step 2 annotation. So let's look at the annotations. I'm just gonna go ahead and configure, edit column, and here I have the list of all my keywords included into my FCS file. And really, I don't have, yes, I do have my Steam keyword, so I can add that Steam keyword, and I think that's it. Okay, so click OK, and I have my Steam keyword, Steam, no Steam already there included. Okay. I don't have any patient ID keyword. If I wanted to add a keyword for patient ID, I can just say add keyword and say patient ID, PID, and can say a one. This one is patient 2, patient 1, patient 2, and patient 1 for the last one. Okay. Usually for keyword, especially for high parameter analysis, I like to put numbers instead of text. So here we have text, we have no Steam until last Steam. That's fine. We have only 2 conditions, so it's going to be easy for us to figure it out. But if I have way more condition, I like better to use numbers instead of text. If you use text, FlowJo, when you're gonna convert those keywords into a parameter, it's gonna use the alphabetic order to put it at the different value. So the first in alphabetic order is gonna be at 1,000. The next one's gonna be at 2,000, 3,000, 4,000, 5,000, etc. So here, 2 conditions. So my first in alphabetic order is my nose team will be at 1,000, and my stimulation second in alphabetic order is going to be at 2,000. If you put numbers, just going to be exactly at the number that you've put. All right, so we're good here. My annotation is done. So now we are in our concatenation step. So let's select all our live lineage negative population. So if it was 4, I could select 1, 2, 3, 4 like that manually, but you're probably gonna have way more than 4. So the best way to do it is select the population you want to concatenate. So here my lineage negative live cells. Go to edit, click on select equivalent node, and it's gonna select all the same population within that group. That's good, that's what we want. Then we're gonna go in file, export concatenate, and we're gonna export concatenate the population. Want here, so it's gonna open the export concatenate platform. We want to concatenate, so we're gonna click on the concatenate tab right there. We're gonna create a new FCS file. It's gonna create this new FCS file on my desktop. Okay, that's fine, that's what I want. If I don't want, I just click and select where I want it. It's gonna here is the downsample step. So right now include all, that means it doesn't do any downsampling. Usually, as I said, we want to do a downsampling just to make sure that we have the same number of events between each condition. So I'm just gonna say include no more than, and the value that's entered here is the lowest value of all your sample, which is this case is 42,497 coming from my TLR team patient 1 right there. And I can put whatever number I want. So I'm just gonna put a really small number today for training purposes, so like that we don't wait too long. So 10,000 event. Then it's gonna ask us, do you want to have the parameter compensated or not? You always want, I would say 99.9% of the time, want all the compensated parameter. Then we're gonna click on the advanced option where you can change the name of this concat file. For today, I'm just gonna leave it as default. And what's most important, it's in this additional parameter right here, I'm gonna add my keyword, and like that's gonna allow me to transform my keyword into a parameter. So go into list, and look for my Steam keyword and patient ID keyword. Okay, and click OK. So now it's gonna create new parameter with my patient ID and Steam keyword. You can spread those keywords to give you a nice Gaussian, so it's gonna look nice in a dot plot, or you can just not spread it. It will look nice in a histogram, but not as good as in the dot plot. So really up to you on how and where you want to present it. For today, I'm just gonna leave it spread. All right, click on concatenate. It created this new concat file that put it on my desktop right here. Okay, and I'm just gonna add it to my existing workspace. Close. All right, you put it in my data here. So I have it in all my sample, but also put it in my data group here. I don't want it there, so I'm just gonna delete from that group. And I'm just gonna create a new group. Create group, concat, like that, make it clean, organized, easy. Okay, this concat group and put my concat file into that concat group. There you go. Oh, I have my concat file created there. You only took 10,000 events for each sample, so that's 40,000 events total. You already selected only the cells that are selected. So for scalar size scalar, it's good. Doublet discrimination, all the doublets will been removed. If we look at my lineage, it's lineage negative, and if we look at my live, then gate is live. Okay. You also created additional parameter. So the first parameter is my sample ID, okay, which is my sample 1, 2, 3, 4 in the same order that my sample were into my workspace when we concatenate. So that means that the first sample is my TLR Steam patient 2, my next one is my TLR Steam patient 1, no Steam patient 2, no Steam patient 1. Careful this order. If you adjust the order here in my workspace, not gonna adjust my order right there. So just be aware of that. But it doesn't really matter because we have our keyword patient ID. Okay, so I have my patient 1, my patient 2, and I have my keyword Steam with my no Steam in 1 and my Steam in 2. Okay, and if I can even do my patient ID versus Steam, oops, patient ID versus Steam, and I can see my full sample. So no Steam, patient 1, no Steam, patient 2, Steam patient 1, Steam patient 2. And can split that back between my full sample or telling, getting based on my Steam, TLR Steam, and no Steam, no Steam. Okay. All right, so I have my concat file. So now I want to do my dimensional reduction. So my dimensional reduction today, we're gonna do PacMAP. So go and plug in, select PacMAP, PacMAP here, select my parameter of interest, so all my fluorescence parameter minus viability and lineage because I already get the cells that were lineage positive and then out of those files, so I don't need those parameter anymore. I can change my nearest neighbor, I can change my mid-near ratio, I can change my FP ratio, and I can change the number of components it's gonna create. So here we're just gonna do a 2-dimension, so X versus Y, that's what I want. Okay, I'm gonna leave everything to default and click run, and it's gonna calculate my PacMAP. Same thing, it take a few minutes to run. while we're waiting, if you have any question, put it in the chat or in the Q&A. You can answer any of those. Yeah, see one. All right. How powerful does your computer need to be to run these efficiently? Um, we, but depend of depend of many things. Depend of how many data you have uh, on that you want to analyze, how many parameter and everything. The minimum requirement for FlowJo, I think we increase it now, is 8 GB of RAM on your computer and an i7 processor. What I recommend is the more RAM, usually the better. I have a 16 GB of RAM on my Mac, and I have an i9 processor on this computer. This computer is 3 years old, so it's not like the best computer that's on the market currently. As long as you have uh, 4 times the amount of RAM on your computer than the quantity of data that you put in FlowJo, you should have plenty. Okay? That's a good rule of thumb on um, the quantity of RAM that you can use, or the other way around, the quantity of data that you can put in FlowJo to run that. That say, all those algorithms, because there's a lot of math involved, so there's a lot of calculation there. So even if you have the most powerful computer, you may have to wait sometimes for at least few minutes, sometimes maybe even hours, depending on how many cells you have um, in your data set. For example, I did one time run a T-SNE on, I think it was 20 million cells. It was more than that, 100 million cells, something like that. And I just put it overnight to make sure that it was running, and I didn't have any issue on blocking my computer running. You can change also the quantity of RAM FlowJo can utilize from your computer, and that's part of your preference files and performance. So you can allow FlowJo to use more RAM. Of course, if you allow FlowJo to use more RAM, that means you cannot run something else on your computer. FlowJo is gonna try to use all the, the power of your computer to run the calculation. So yeah, it's really up to you. All right, so PacMAP is done running. So we have here the visualization. We have on contour plot. If I want to put it on pseudo color, I can. I can smooth it up if you want, whatever visualization you want. So you can see different islands, good, different cell population. All right, so our dimensional prediction is done. Now we're gonna run our XShift to do our clustering. So we go here, XShift, same thing, select my parameter for XShift. And you see the k-nearest neighbor changing depending on the number of parameter I'm selecting. Okay, so for example, if I remove variability and lineage, I have 102. If I put them on, to have 82 as a k number. So he automatically finds that k number for you. As I said, you can change it, but usually the default is pretty good. Because I have more than 10 parameter, there is going to be the angular distance metric. If I had less than 10 parameter, it will be Euclidean. Um, I'm gonna leave at 40,000 event here on the downsampling for XShift, and I'm gonna leave out a run ID automatic. I can put whatever value I want here. If I want to put a special value, here in this case, I leave it automatic. So XShift is running, and it's gonna give you, I think, 12 cluster for this data set. Not 100% sure, but something like that. Yeah, there you go. and I did run XShift, and I have my 12 cluster created. Okay, so I did my dimensional reduction, I did my clustering. So now we're gonna do the phenotyping. So on phenotyping, I can be manual, as I mentioned into the slide. So I can go into my workspace tab, algorithm, and I have Cluster Explorer. Click on it, select first the clustering algorithm I'm gonna use. I only run XShift today, so I'm going to display XShift. If I run many, I will see all of them available and select which one I want to display. Maybe I want to display many, so I can select multiple there. But today is only XShift. What parameter I want to display, so everything but my lineage and variability. And what dimensional prediction I want to display, my PacMAP and what I run. So click on accept. Okay. So I have my PacMAP visualization right there. I have my, sorry, Zoom is in the way. I have my cluster size right there. So number of my cluster number and the percentage of cells for in each cluster. If I want to have the actual number of cells, I can select that and go in the option and remove the normalization. I can see the actual number of cells. I'm gonna leave it on percentage. I have my control panel here. I have my graph line. So basically the MFI for each marker for each cluster. So if I take, for example, cluster 1, I will see the expression of IL6, HRDR, CD1C in for the cluster 1, and I have my heat map. Okay? So here is really interactive, so I can really click on cluster 1 and see the expression of cluster 1, same thing for cluster 2, 3, 4, 5, 6, 7, and you can see, for example, 6 seems to be positive for a lot of things, HLRDR, TNF, CD83, CD14, IL-1 beta, negative for CD16, positive for IL-12, and negative for CD123, negative for IL-10, so CD14 positive, so monocyte, CD83 activation marker, so activated monocyte, and we see some cytokine expression with TNF alpha and IL-1 beta. So I can rename my cluster 6 as activated monocyte, for example. Okay. Um, I can gate also. I can select, for example, only what's CD14 positive, so create a gate so I can see like there's 3 cluster, the CD14 positive, the number 5, sorry, number 6, number 10, and number 11. I can see the visualization on the PacMAP where they are located. Right now in my graph line, only see the mean fluorescence intensity. If I want to see median, I can select median. That's up to you. We can do an AND gate or a NOT gate. So I want to do an AND gate, and I want to see what CD14 positive and HLRDR positive, and now I see the double positive. And now I'm gonna create a NOT gate, but I don't want to see what CD16 positive, so I'm just gonna go a gate around the CD16 positive. So I see DR positive, 14 positive. Um, That's not what I wanted. I said 16 negative clear gate. We do that. So uh, DR positive, 14 positive, and not the 16. And I can see that my cluster 6 and my cluster 11 match those criteria. So the only difference between those two cluster is by the heat map or graph line. I see one is TNF alpha positive, the other one is TNF alpha negative. One is IL-6 positive, the other one is IL-6 negative. Um, Same thing with IL-1 beta, and for the rest is the same. So it's probably activated monocyte and non-activated monocyte. Okay. All right. So really I can rename all my cluster like that one by one, looking at my graph. Of course, all those graphs can be copied, so you can copy those image and paste those image in uh, any thing, so PowerPoint or whatever you want, so copy image, paste it. But anything that have a number, like your cluster size or your heat map, you can copy the content and paste those into Excel. So to have the actual number of a number of cells or percentage, or here the MFI for each cluster and each parameter for the heat. All right, so now we define all the different population. I can also define this population using the mem. Let's say you just do a mem score, create mem score, and it's gonna ask you to select the parameter. And I also put the threshold, population of reference, either the auto reference, or it's one cluster versus all the other, or the user define mem. Okay, I'm just gonna leave it like that, the default, because I don't have population reference. I see there's a question. How do you assign a name of the cluster? Or you can just go back here for cluster 6 and just right click, rename, and name that cluster. Okay, so cluster 6 is activated on the side, so I can rename it whatever I want. All right, so we have all the naming. Maybe this naming is long. A lot of parameters. So I'll show you that you can put a threshold. So we can put a threshold of 2, for example, to see this list being shrinked. And our cluster number 6, we see that is CD14 positive, vector of 2, and IL1 beta, vector of 3. So we saw that it was activated monocyte when we look at the cluster 11, it's only CD14 positive, DR positive, so just my regular monocyte, non-activated monocyte. Okay. All right. Now we want to do the comparison. Go back to Cluster Explorer. Oh, I see another question. Yeah, so no, the naming is not in the cluster interface. So, but you can have Cluster Explorer open and just right click and rename it when Cluster Explorer is open. But not it's not in the interface. You can have the naming after in FlowCut, after you rename all those cluster with like your biological name, instead of having cluster 1, 2, 3, 4, 5, all the way to 12 for us today, you can have the actual name that you put here into your workspace. But no, the naming is into your workspace. Right, move that. Let's just focus on Cluster Explorer. Tuk, tuk. All right, so now that we want to do the comparison, so we saw that on the workspace we already created 2 different populations based on the stimulation, so no steam and steam. Okay, so now we're gonna use this getting that we have done right there in Cluster Explorer. So you can do it here instead of visualizing the full concat file. You can only visualize the cells that are coming from your no-steam patient. You see that my PacMAP changed. See that we lost one of the island. You can see that the frequency also changed, and same thing for the TLR. So you see that here in my TLR steam, I get mostly my cluster 11, but lose the thing here on top, and vice versa. I think I inverted those two naming, by the way, but it doesn't matter. You get the idea. All right, so go back here, and we can also add additional node in your heat map. Okay, so that's one way. Zoom in the way, make me click the wrong way. Okay, so we can go in option, add a node as column, okay, and add our two condition, no steam, steam, click OK. And now we have the color coded, and we see that whatever is green, that means no change or little change between those two conditions. Okay, a little bit more cells coming from my TLR steam condition for my cluster 1 to 5, and a little bit less from my no steam to create those clusters, but not a big change. You see in the scale, whatever is green, we are around the 50%, or maybe I go to 60-40. If I put my mouse over, I'm gonna get the exact numbers, so 74 and 26. Okay, and whatever is blue here, that means it's almost no cells. Exactly 0% here on my cluster. What's cluster that? Cluster 8, no cells in my condition, TLR steam condition. 100% of the cells to create this cluster are coming from my no steam condition, and vice versa here on my cluster 11. No cells, or like 2% of the cells that create that cluster coming from my nose team, and 97.9% cells that create that cluster coming from my keyword TLR steam condition. Okay, so we giving me which as cluster is on which condition. All right, and with that, as I say, we can export all those images. And with that, that's the end of this training. Happy to take any additional question that you have. Right. Yes, FlowJo does not work on Linux. You're correct. However, the R package Flow Workspace from RG Lab say it can replicate the gating from FlowJo using the FlowCut functionality. I'm not aware of that, so I cannot answer your question. I don't know. I don't know about that R package. Never, didn't try it. Yes, this webinar is recorded. It will be put on our website. So if you go on our website, on flowjo.com, you go in the Learn webinar. Here you have the list of the live webinar, but if you go at the bottom, you can see the recorded webinar, and you will see the list of all the recorded webinar. We keep usually the past 2 years of the webinar, sometimes a little bit more if we didn't create a new webinar to replace a previous one. So in the next few days, you should find it over there. Have each clustering algorithm scale up with the number of events. You can put as many events as you want in your clustering algorithm after it's just a number of time, how long you're willing to wait. Or you can put millions of cells. It's not gonna give you a better clustering. It's just gonna take more time to give you the results. But if you have a really rare population, make sure that you have enough cells for the algorithm to find them. So it's a 0.01%. Know that you need to have at least I will say 50 cells plus, yeah, to for the algorithm usually to be able to define that as a cluster. So do the opposite in the math. All right, to download also those plugins, so just go on our website, flow.com, click on the exchange, okay? And here you can just display the one for FlowJo, and we can look for FlowCut, okay? And the FlowCut, you can download it, or you can XShift, there you go, Shift there, and if you want PacMAP, you know, download it there, okay? What's the difference between the export concatenate button and downsampling plugin? Um, just we updated the export concatenate button, so it's the same basically. Here with the export concatenate platform, now you can do the downsampling with it. Before you had to add the plugin, the downsampling plugin to do it. So faster, easier than using the platform, the concatenate platform, than just using another plugin. But we use a similar method of downsampling for both. Oh, you're welcome. Any more questions? Yeah, so for the question earlier, so I did answer already. Um, so what marker need to be selected for running your dimensional reduction? Basically, all your marker except the one you get out, you get it out. So for me, it was my lineage and live dead marker to define my population. If you define your population with your CD45 marker before you concatenate, for example, you remove the CD45 marker. So everything that you already use for your manual gating, just remove it. So, and to determine the number of cluster, well, XShift find it for you. So you don't define the number of cluster, only flow sum. You need to put the actual number of cluster that you're expecting. XShift or Phenograph or T-Rex automatically gonna define um, that number of cluster for you. After that number of cluster may not be the one, may not be able to define all the population. Maybe you have more population than the number of clusters that's been created, or vice versa. So you're gonna have the number of clusters gonna be too much compared to the number of population that you have. But it's better to over cluster than under cluster. So for flow sum, that was your question. Um, to know the number of cluster, that's the tricky part for flow sum. So if you don't know the number of cluster that you have and you want to use flow sum, you can just put a big number. As I said, it's better to over cluster, and after you can merge a cluster that are the same cells. A good rule of thumb that I usually use when I don't know the number of population is I put the number of marker I have in my panel. So if I'm running a 20 color experiment, I put 20, um, 20 cluster. And hopefully that's good enough to define all my population. If it's not, if I do my cluster explorer and I look into it and I see that I have more population than that, I can rerun flow sum with higher number. But usually does a pretty decent job. And after you have tools also to evaluate your clustering algorithm. One of the tools is called Euclid. So you can look at Euclid to see if really each cluster is only one population or if there's uh, two population or more into that cluster. So Euclid is a plugin and it's right there. Measure the quality of clustering. All right. So if there's no more question, I'm gonna let you go back to your work.